Welcome to the second module on zebra and quagga mussels and other ANS biology. We will review the top species in Colorado, including the most recent information on ANS detections in Colorado. The species we will focus our time on are those which have been detected in Colorado: zebra and quagga mussels, Asian clams, water fleas, New Zealand mud snails, rusty crayfish, and Eurasian watermilfoil. Fortunately, the watercraft inspection and decontamination protocols in place for zebra and quagga mussels will work to prevent the spread of other ANS as well. Remember, no mud, no plants, no water, and no animals on boats, no exceptions. It's important for inspectors to learn the basic biology of invasive species so that they may understand the science-based approach to prevention, containment, and monitoring in Colorado, as well as to answer questions from members of the public. Let's begin with the poster child for our program, zebra and quagga mussels. As you'll remember, they have 3 life stages: veligers, settlers, and adults. Veligers are microscopic and can be transported in standing water. Settlers are the juvenile life stage that can appear as sand or bumps on boats that don't rub off. Adults are the mature, 2-shelled organisms with Bissell threads that are more easily identified. There are 3 main characteristics that make zebra and quagga mussels invasive. First is their ability to reproduce at alarming rates. Second is their ability to attach to surfaces with Bissell threads. Third is their ability to filter feed. Let's review each one of these invasive traits. Zebra and quagga mussels are extremely good at reproduction. One female mussel can produce up to 1 million eggs per year. If only 10% of these survive, there would still be 10 septillion mussels in the water body after 5 years. Under the right conditions, mussels can produce multiple spawns per year. In Lake Mead and the lower Colorado River, they are reproducing monthly. This means that there are so many organisms in the water body, they cover the bottom, sides, and shorelines quickly. In doing so, they displace native species, damage equipment, harm infrastructure, stop the flow of water, and ultimately make it very hard for boating or other aquatic types of recreation to occur. This type of infestation would be devastating for Colorado's parks and wildlife. Bissell threads are another characteristic that cause zebra and quagga mussels to wreak havoc on ecosystems they invade. There are no native species that have Bissell threads in Colorado. These spiderweb-like threads are made up of proteins that are secreted from a gland inside the mussel. Bissell threads allow zebra and quagga mussels to attach and smother native organisms, damage equipment and boats, and impede the flow of water to homes, farms, power plants by clogging pipes. All these mean bad things for natural resources and costly long-term repairs to infrastructure. The third invasive characteristic is their ability to filter feed. One mussel, the size of your pinky fingernail, can filter up to 1 liter of water per day, removing the base of the food chain and any other materials in the water column. Zebra and quagga mussels are one of the only species able to crash the food web at multiple layers, smothering benthic organisms and removing the base of the food chain, plankton. An established population of adult mussels in a body of water can filter out a majority of the nutrients in that body of water, initially making the water clearer. But Colorado's waters aren't meant to be clear like the Caribbean. Clear water is a bad thing for Colorado because it means that the water body is lacking aquatic life and is not biologically productive, which is not good for our fisheries. Once the water clears, sunlight can penetrate the depths, and over time, aquatic weeds can form dense mats that impair recreation and water storage. Once there are dense weed mats, algae blooms begin to occur on the surface. Oftentimes, these weed mats and algae growths cause a reduction in oxygen and prevent aquatic life from functioning below the surface. Growth of nuisance algae can also cause toxic water conditions, killing fish, polluting drinking water, and cause a foul odor. Another invasive species often thrives from these conditions: West Nile virus resulting from too many mosquitoes and impacting human health. All of these negative secondary and tertiary impacts from mussel invasions. Management of mussels is very difficult and expensive. Eradication is impossible in open water systems such as western reservoirs that have a river running through them. Prevention is the best and most cost-effective choice. Education is the most important part of prevention. If boaters know that they can protect their waters by cleaning and drying their boats, we can greatly reduce the likelihood of introducing ANS. The only pathway of introduction into Colorado is on watercraft. Now that we've learned why we don't want mussels in Colorado's reservoirs, Where do you think they are most likely to be found on boats? Rough areas, right angles, dark or shaded places, moist or wet places, below the waterline, and in compartments that hold water. Attached is a recent national map of mussels from USGS. You'll notice that Colorado's locations are now listed as locations where we have delisted mussels due to 5 years of negative detections. There are no positive locations for zebra mussels in Colorado. The only positive water for quagga mussels is Lake Pueblo State Park. We've never found an adult in Colorado. We've only found veligers. The last detection in Pueblo was in 2011. That means if we don't find any veligers or adults in 2015 or 2016, we will delist Pueblo to a negative water in 2017. For now, Pueblo continues to implement the strict quagga mussel containment procedures, which includes high-risk mandatory exit inspections and standing water decontaminations for boats that are not returning on exit. This is stricter than the new other ANS positive water containment being implemented this year. In 2015, Pueblo will be piloting a new receipt. You'll learn about all of this in depth in the next module on inspection. Next, we'll talk about the species in Colorado that is most commonly mistaken for zebra and quagga mussels by both customers and professionals alike, the Asian clam. There are a couple of characteristics that can help you tell them apart from zebra and quagga mussels. Asian clams do not have Bissell threads and therefore cannot attach to substrates like zebra and quagga mussels. If you run your finger across an Asian clam, you will feel very deep ridges in the shell. Zebra and quagga mussels have smooth shells. They're also not striped like zebra and quagga mussels. The striped appearance of the clams in this picture are due to sun bleaching the shell's ridges. In this photograph from the shoreline at Lake Mead, you can see the differences in size and shape between quagga mussels and Asian clams present in the rocks. Like zebra or quagga mussels, Asian clams are small, bivalve mollusks that are not native to Colorado. They were first found in the South Platte River drainage in 1993 and are now known to the Arkansas, Colorado, San Juan River basins, and South Platte River basin. Because they have a similar life cycle to zebra and quagga mussels, they can also be transported in water, and juveniles can be transported in mud. They cannot attach to the hull of a boat because they do not have Bissell threads. While they are somewhat widespread in Colorado, it is still important not to move them into new water bodies. The inspection protocol will prevent the spread of Asian clams in Colorado. Adult Asian clams have an oval shell that is deep on the hinged side. They can grow up to 65 millimeters in length. However, the average size is 25 millimeters. The shell has well-defined, thick growth rings that feel like ridges or steps on the shell. Older clams are darker in color, but all vary in some shade of brown. Scientific literature says they prefer fast-moving water, but they are commonly found in lakes and reservoirs in Colorado. Veligers can easily be transported in standing water and by the natural flow of water. They are also moved around as bait, cultivated for food in Asian markets, and also sold in aquarium trade, falsely labeled as pygmy clams or golden clams. They are prolific reproducers, and huge population numbers can clog intakes for water canals, power plants, and irrigation despite not being able to attach. Similar to zebra and quagga mussels, Asian clams outcompete native bivalves for food and space, disrupting the aquatic food web. Next, we will discuss the water flea, Daphnia lunholtzi. This is the species most recently detected in Colorado. Water fleas are aquatic zooplankton crustaceans that have a jumpy or jerky mode of swimming. The Daphnia water flea were introduced from the aquarium trade and as contaminants in fish stocking. They are native to Africa, Asia, and Australia. Unlike the fleas dogs are known to carry, water fleas are very different. They do not live outside the water and do not bite or harm people or pets. Daphnia lunholtzi has a unique body shape and is distinguished from other zooplankton by its long tail, about the length of its body. The head is mostly a single large black eye, and they have a swimming antenna that propels them through the water, allowing travel between shallow and deep waters. Water fleas can range up to 3 millimeters in length, depending on sex and age. A microscope is needed to positively identify Daphnia. When a fish predator is nearby, the non-native Daphnia lunholtzi will detect the fish hormones and deploy an elongated helmet, elongated tail, and additional abdominal spines to protect itself. When the helmet and long tail is not present, the invasive lunholtzi will look like the native Daphnia. However, native Daphnia species do not have these defensive characteristics. Water fleas have both sexual and asexual reproduction capabilities depending on environmental conditions. It takes less than 2 weeks to start a new generation with asexual reproduction. Resting eggs can lie dormant for long periods of time. Resting eggs can also pass unharmed through the digestive tracts of fish. Eggs in adults are easily transported in ballast tanks, bilge water, bait buckets, live wells, on fishing lines, anchor lines, and nets. It only takes one adult or egg to start an infestation. If female water fleas die out of water, under certain conditions they can produce eggs that resist drying and freezing, which can also establish a new infestation later. They outcompete native juvenile fish for food, causing low survival rates, and because their tails stick in the throats of juvenile fish, they are unpalatable. Their long spines can cause them to become entangled on fishing lines, and they can clog the eyelets of fishing rods. Daphnia lumholtzi were first identified in Colorado in Pueblo Reservoir in 2013. The CPW Aquatic Animal Health Lab has identified 14 total locations in 2013 and 2014. This is a map of the known water flea locations in Colorado. Almost all of these waters are WID sites, which will now be considered an other ANS positive water, and will be using blue receipts in 2015. Now let's take a moment to review some information on New Zealand mud snails. New Zealand mud snails were first found in Colorado in 2004 in 11 Mile Canyon. These freshwater aquatic snails are native to New Zealand and were first discovered in North America in the 80s in Snake River of Idaho and the Madison River in Montana. While there is a native snail that looks similar to the New Zealand mud snail, there are some features to help identify it. It's very small, ranging in size from a grain of sand to 1/8 of an inch in length. They are dark brown to black in color and have 5 to 6 spirals on their shell. When the tip is pointing upward, the opening to the shell faces right. Most native snails open to the left. They can close their shell with an operculum and protective membrane and remain protected in damp conditions for up to 30 days or on damp surfaces for up to 50 days. Like all invasive species, they outcompete the native species for space and food and reduce the availability of prey for fish. They are not a food source for fish because they can close the opening of their shell with their operculum and protective membrane and pass through the digestive system of fish unharmed. This helps spread the snail and results in smaller fish due to lack of nutrients. The fish think that they are eating snails, but they are just moving through the fish's digestive system and coming out the other end happy in a new home. This is the list of where New Zealand mud snails have been found in Colorado. There were no new detections in Colorado in 2014. We had a very aggressive angler education campaign in 2004 to encourage anglers to clean gear, primarily waders, to prevent the spread. In 2008, we turned our attention to boaters, and New Zealand mud snails began popping up in new places annually. We have been working over the last year to get back in touch with anglers and educate them as to the importance of cleaning their gear. Waders are the primary vector of New Zealand mud snail spread. This is a map of all known New Zealand mud snail locations in Colorado. The reservoirs with WIDs that are positive for New Zealand mud snails, 11 Mile and Spinney State Parks, are both considered other ANS positive waters and implement containment with blue receipts. Let's move on to another ANS, rusty crayfish. They are native to eastern USA in the Ohio River Basin and were initially discovered outside of their native range in the 1960s. This aggressive species has had devastating ecological impacts on the northeastern U.S. Nativity can be quite local. The species is native to the U.S., but not to Colorado. We have 6 species of crayfish native to the eastern half of Colorado that are not native west of the Continental Divide. In fact, there are no native crayfish west of the Continental Divide. All crayfish west of the divide are there because people have moved them there. Identification of rusty crayfish in the field is extremely difficult. Rusty crayfish grow up to 5 inches long and have brown bodies. To positively confirm the species, it must be a reproducing male specimen and is confirmed by at least 2 experts. Some characteristics of rusty crayfish, which may or may not be visible, are 2 rusty patches on either side of the abdomen, an oval gap in the claw, and black bands on the tips of the claws. Rusty crayfish will colonize freshwater lakes, rivers, and streams, and prefer deep pools and fast cover from predators. These crayfish were introduced into Colorado through illegal stocking and bait movement. These crayfish are voracious eaters and will prey upon small fish, insects, and fish eggs. They will also eat aquatic vegetation and move plant communities as a result. This affects the food chain, reduces cover for prey fish, and reduces the available food for wildlife. In turn, they are not good fish food themselves, as they are extremely aggressive and will fight off fish. They also have a thick exoskeleton, making them difficult for predators to digest. It is reported that popular recreational swimming holes in Wisconsin and Minnesota have been ruined because the rusties will pinch swimmers. Rusty crayfish are very limited in distribution in Colorado. They have been identified in Catamount Reservoir, the Yampa River south of Steamboat Springs, and Stagecoach Reservoir in Routt County. They have also been found in Sanchez Reservoir in Costilla County in southern Colorado. CPW controls the population through mechanical removal in an attempt to mitigate impacts of fish populations and the public. This is a map of where rusty crayfish have been discovered in Colorado. There have been no new detections since 2011. There is only one WID station positive for rusty crayfish, Stagecoach State Park. While considered in other ANS positive water, they are not implementing containment rules because rusties are being managed through a must-kill regulation, as well as intensive manual removal and education efforts. Rusty crayfish cannot be transported in standing water, and the exit protocols in place for prevention waters will detect them if they are being illegally transported live inside a bait well or other compartment on a boat. It is against the law in Colorado to move live crayfish anywhere west of the Continental Divide. Again, there are no native crayfish in Colorado west of the Continental Divide. Live transport is also prohibited from Sanchez Reservoir, where rusty crayfish are known to exist. If any are caught by anglers, they must be immediately returned to the body of water they came from, or killed by removing the head from the thorax prior to leaving that body of water. Now let's discuss an ANS that is also a noxious weed, Eurasian Watermilfoil. Eurasian Watermilfoil is present in several locations throughout the state. The most recent detection in 2014 is at Chatfield Reservoir State Park, in addition to water fleas. This plant is native to Europe, Asia, and northern Africa. It was most likely introduced through the nursery trade in the 1940s, but possibly as early as the late 1880s. This is a highly aggressive species that can survive in a variety of habitats and grows an average of 1 foot per week. These dense mats crowd out native species, disrupt the food chain, and displace native wildlife. It also impedes water recreation such as boating, swimming, and fishing. Eurasian Watermilfoil is a submerged, rooted, perennial plant. It has long, branching underwater stems with feather leaves in whorls of 4 or 5. Leaves are in pairs of 11 to 21 that are closely spaced. The leaves are about a ½ inch in length with a blunt tip. The plant has no structure and will collapse out of water. Here are some pictures of Eurasian Watermilfoil for your reference. Please report Eurasian Watermilfoil if you see it at a site that is not already known to have it present. Eurasian Watermilfoil is not widespread in Colorado. Most locations are on the Front Range, from Fort Collins to Pueblo. There are only 2 locations on the west slope, and are in the Rio Grande and Navajo Reservoir in the southwest. The majority of the west slope and eastern plains are free of this invader for now, and we're trying to keep it that way. WIDs with Eurasian Watermilfoil are now considered other ANS positive waters, and will be utilizing blue receipts for containment in 2015. However, if you go back a few years and compare today's distribution with that from 2012, you can see that Eurasian Watermilfoil is continuing to spread to new locations along the Front Range. New locations in the past few years include major water bodies such as Chatfield, Cherry Creek, and Pueblo. As a result, Eurasian Watermilfoil and its aggressive hybrid continues to be a concern for water and recreation managers in the state. For your reference, this is a list of watercraft inspection and decontamination stations with a known ANS population. Red stars indicate new detections in 2014, mostly water fleas and Eurasian Watermilfoil. While Pueblo continues to be the only site to implement the ZQM containment procedure, the rest of these locations are now considered other ANS positive waters and will be using blue receipts. You'll learn more about the new protocols for these sites in the next module on inspection. Here's a map of all known ANS in Colorado. As you can see, much of our great state is still free of ANS, and your hard work will keep it that way for future generations. This concludes Module 2 on biology. Please take the Module 2 quiz and proceed to Module 3.